All right, we're going live. Oh, we're live now. Okay. Hi, and welcome to Expanding Horizons. My name is Gabrielle Gutierrez. I'm currently at the University of Washington, and I will be moderating this session with Eva Dyer from Georgia Tech. We are excited to have an incredible lineup of speakers at many stages of their careers. The goal of this session is to highlight underrepresented voices and new scientific perspectives here at Cosyne. Each presenter will give a 15-minute talk about their research. We will take a few questions from the audience after each talk, so please enter your questions into the Slido tab, and one of the moderators will pose them to the speaker at the end of their talk. Our first speaker is Ilenna Jones. Ilenna is a PhD student at the University of Pennsylvania in the Coding Lab. Today, Ilenna will tell us about her talk entitled, Might a Single Neuron Solve Interesting Machine Learning Problems Through Successive Computations on its Dendritic Tree? Go right ahead and start sharing your screen, Ilenna, please.
Wait a second. And hello, everyone. So my title is, due to a bit of a last-minute mishap, my title is an old title. The title of this talk is Might a Single Neuron Do Interesting Machine Learning Problems on Successive Computations of its Dendritic Tree. The old title is Can Single Neuron Solve MNIST, the Computational Power of Biological Dendritic Trees? So before I get into that kind of question, I have to start with something a bit more basic, which is, what can a neuron do? You can define the word do in terms of role. For example, what do neurons do to mediate behaviors? If you were to have an auditory circuit made up of neurons and you removed one neuron, you might not be able to hear anymore. Then you might say that it is an auditory neuron, and thus you're assigning a role. That's a form of do. Another form of do is to define it in terms of computational capability, wherein you might consider a neuron as an input-output device where it receives inputs in its dendritic tree, integrate those inputs into the soma, and outputting an action potential spike. In this talk, I will be discussing the output of a neuron as simply a binary output, which is the presence or absence of an action potential. So we'll be talking about what can a neuron do in terms of computational capability. So we can approach a neuron as an input-output device, and we wouldn't be the first. In 1943, McCulloch-Pitts made the McCulloch-Pitts neuron, which is a linear weighted sum of its inputs put through a threshold nonlinearity to produce a binary output, 1 or -1. And if you hook a bunch of these McCulloch-Pitts neurons up in a chain, you can get logic functions. Fast forward 70 years and more, you can take many neurons of this type, where the activation function is a little different from this, and take thousands of them, and you can get an artificial neural network that is able to do complex, high-dimensional image classification tasks. I say high-dimensional because if you were to take an image, a pixel of that image is a dimension. So this pictured here below is AlexNet of fame, 2012 fame. So both McCulloch-Pitts neurons and artificial neural network neurons are linear weighted sums of inputs mapped through an output nonlinearity. However, this assumes that dendrites are linear and that their inputs are linearly summed. However, we know that dendrites are nonlinear. They can do illogical operations. They have phenomena such as coincidence detection, low-pass filtering, attenuation, and more. So point neuron models, despite their success, may be grossly underestimating what neurons could do. So how do we ask what neurons can do? I'll be talking about two general methods for approaching this question, one of which is a more traditional method and one of which is an experimental method that I'll be using for this project. So the more traditional method is the empirical neuron modeling procedure. So if you're going to take an input current, give it through an electrode to a real neuron, and then record its voltage trace to, in this case, we're only caring about the action potentials, you would then get an input-output function of a real neuron. Then what you want to do is you want to take a neuron model of your choice based off of the questions you're trying to ask and fit that neuron model to the input-output function such that if you provided the input to this neuron model, it would produce an output that would match the real neuron output. This is very useful for testing counterfactuals, and a neuron model that is fit in this way is considered a good neuron model. However, there's a second way we can approach this question of how do we ask what neurons can do, and this method, this other method is inspired by that of artificial neural networks. Artificial neural networks also fit input-output functions, but those input-output functions are called tasks. Some of them even image classification tasks, where the input may be a very high dimensional image and the output is an image label. So what we want to ask then is, like with artificial neural networks, can we test a neuron's ability to learn to perform tasks in this way? So if we put these two methods side by side, the empirical setup and the task-based setup, instead of a somatic current or a very low dimensional input, you can get a many dimensional input of a dendritic task inputs. And then for its output, instead of an empirical recording of what a real neuron is doing, you have what you impose as the correct classifications of the task inputs. And across these two methods, the neuron model can be kept the same. The reasons why we might do this is so that we can take a principled approach to setting what the dendritic inputs are, and we can quantify neuron model performance via an accuracy measure, just like we do with artificial neural networks. So what exactly is the input-output function that we're interested in? The type we use is a binary classification of images. Binary in that, if you remember what I said about output, we care about the presence or absence of an action potential, which is a binary output. So we care about binary classification. If we look at the top data set, which is MNIST, we have 7 data sets here. Here are pictures of MNIST. There are handwritten pictures of numbers. And it's a 10-class data set from which we chose 2 classes that were the most confused when set next to each other in comparison, and by a linear classifier. And these input images are 1,024 elements long. That means that there are 1,024, our neuron model would have to receive 1,024 synaptic inputs. And we do this for 6 other data sets. So what does our neuron model, which is receiving these inputs, even look like? Our dendritic neuron model we can dub the one tree, and that will become clear in just a second. So if you look at this cartoon of a neuron with its dendritic tree, one could even say that this looks less so like a pyramidal cell, but more so like a stellate cell. You can see that it has several subtrees, and on one subtree, it's receiving a 4-dimensional input, 1, 2, 3, 4, on its tree. Now, what makes this so, this in any way dendritic is the binary tree structure of the tree, where 1 and 2 would be received by a downstream section of the tree, and only 2 are received. Same with others, until you get to the soma of the neuron. If you want to retain this biological property of binary tree sparsity, you can do so with an artificial neural network, which we call the one tree, where each of these blue nodes is the input, the white nodes are the dendritic tree branches or compartments, whatever you'd like to call them, and the last node, the pink node, being the soma node or the output node. And the white and pink nodes have nonlinearities involved in them. So we want to understand how well this one tree performs a binary classification task, and we can judge that performance via accuracy. How do we know that the one tree is performing well or poorly? We can compare it to a lower bound, which is a linear point neuron model approximated by linear discriminant analysis, a lower bound, and an upper bound, which is a fully connected neural network that is densely connected as opposed to sparsely connected. This is a two-layer neural network, which we expect to perform better than anything with sparsity. Those are expectations. So the data that we have largely meets this expectation. If you look in A, the x-axis has the one tree LDA and linear classifier and the fully connected network, and the one tree performs better than the lower bound and below the upper bound, which is also replicated in multiple other data sets, but not all. And one thing that we can conclude from this is that this artificial neural network, made to be more neuron-like through its dendritic binary tree constraint, causes it to limit its performance on these tasks. So what else can impact single neuron computation? What other biological properties might we consider? We know that neurons have thousands of synapses, and of these synapses, a presynaptic axon can synapse on average about 4 times per postsynaptic neuron. And this means that a synaptic input can be repeated on average 4 times on the postsynaptic neuron. In vitro multisynaptic batons, which are also doing these synaptic repeats, onto the same dendrite increases approximately 6-fold after LTP induction protocol, meaning that learning might also impact the replication of synapses, which leads us to the hypothesis that repeated synapses may also impact single neuron computation. So we modify the one tree to be a K tree, where K is the number of inputs, and you can see the artificial neural network form of this here, and the inputs are repeated identically to each subtree. And our lower bound doesn't need to change, but our upper bound does in order to compare to this. And so we do so by making the fully connected network match the K-tree parameter size, where we take the hidden layer node of the fully connected network and make it twice the number of subtrees in the K-tree, which leads us to the following results. So this is a bit of a complex slide, but we'll just focus on A. If you look at the x-axis, we have the different architectures where K is listed here, 1, 2, 4, 8, 16, and 32, and the linear classifier labeled here in gray. And the gray line is the fully connected network performance or accuracy, and in black is the K-tree performance. And as you can see here, the K-tree, as K increases, their performance increases. And also the gap between the K-tree and the upper bound fully connected network actually narrows as you increase K. And this can be seen in multiple other data sets. And even in the case where in the fashion MNIST, eMNIST, and USPS data sets, the 32 tree, or the K-tree where K is equal to 32, is indistinguishable from that of the performance of the fully connected network. Some discussion. So there are several limitations and assumptions with this work. A one-dimensional or a vectorized image classification task puts our neuron model out of brain context. It's not likely that a neuron is going to get an ordered vector of pixels as its input. Our model is a tenuous approximation of a real neuron. Neurons are not feed-forward like our model. Synapse location is not limited to the terminals of the dendritic tree like our model. And our neuron model is receiving real-valued inputs, not binary inputs. But with all of these limitations and assumptions acknowledged, that allows us to be very clear on what our results provide insights on, which is the biological properties of dendritic tree structure constraints in repeated inputs. Some relevance to deep learning is that the sparse network can perform at the level of the dense network using nearly the same amount of free parameters. Future work would be to compare K-tree sparse network to other kinds of sparse networks, like Prune's sparse networks from the lottery ticket hypothesis paper and so on. We bring a new approach. So we consider bringing deep learning approaches toward neuron modeling, where we judge neuron model performance instead of traditionally trying to match neuron physiology data. And our results suggest that repeated inputs to the dendritic tree can improve its ability to perform complex computation. In addition, the dendritic tree constraint, which is not listed here, but the dendritic tree constraint also seems to limit its ability to perform these complex computations. Some future work is to introduce biological constraints, more biological constraints, and train these biophysically realistic neural models to do image classification. With that, thank you very much to the Corting Lab, the Neuroscience Graduate Group at the University of Pennsylvania, as well as the Howard Hughes Medical Institute, which funds me. With that, are there any questions?
Excellent. Thank you so much. Yeah, we have a few questions here. So we have one question from John Mastola. So he asks, reminds me of design principle style arguments for gene networks. What are their insights about network structure do you think this approach could provide?
Yeah, well, one thing that was brought up in one of the audiences was how this is kind of like almost like a winner takes all assembly type of thing. So if I go back to, sorry about flipping through all of the slides, but say that each of the subtrees of this K-tree is in assembly, and they all kind of come down to the single soma node, and perhaps each of these subtrees are dealing with the identical inputs differently in their interaction allows it to produce a final output that correctly or incorrectly corresponds to the classification label. But otherwise, I'm not really sure what I could say about the gene network thing. I think I'd have to look into that further.
So we have another question here. How does learning speed versus end performance compare for the dendrite model versus the matched ANN model?
So could you read that again?
So they were asking how the learning speed compares for the two different models. So kind of, training time and efficiency and those sorts of.
I don't have figures for that right now, but what I can say is that the fully connected network, it trains much faster than the K-tree, and it takes much fewer epochs to train than the K-tree. And actually, I think the one tree takes less time to train than 32 tree, for example. So it's not necessarily going to beat those benchmarks, machine learning benchmarks, but that's not really the point. But that's okay. It's the learning for the fully connected network is still much better.
Excellent. Thank you so much. We're going to wrap up here and move on to our next speaker. Thanks again for sharing your work with us, Ilenna.
Thank you.
So up next, we have Fernanda Matias. Great. Hi.
Hello.
So Fernanda is an assistant professor at the Universidade Federal de Alagoas in Brazil. And today, Fernanda will tell us about her work. Statistical complexity is maximized close to criticality in cortical dynamics. Take it away.
Okay, so can you hear me? Great. First of all, I would like to thank all the organizers of this amazing event, and especially Eva and Gabrielle for this workshop that I think it's really necessary to promote diversity in computational neuroscience and science at all. So I will talk about some kind of new results from our group that I think they are quite nice. And it's about complexity and criticality. So I will try to define both concepts if you are not used to it. And then I will show how we connected them both in experimental data and in model. So first part, let me talk a little bit about complexity. So in physics, we typically define the systems or characterize the systems from regular to random using entropy measures. So in such a way that if we have something that is very regular, a regular structure, a crystal or something like that, like this figure here, we would have entropy equals 0 or a minimum entropy. And in the other side of the disorder scale, we would have something that is random. Like if we have a box with something that can be broken like a glass and we shake it, and then after all we destroy everything, we have this random structure and the entropy is maximum or is 1. But everything in between, how can we measure what's going on? So we can have the brain, we can have Van Gogh, or a lot of other complex systems. And we would like to characterize these systems using something extra than just the entropy. So if we can think about a complexity measure, something that we can quantify in these complex systems, we would have something like a small entropy for regular and high entropy for random. But these two kind of boring systems that are very regular or very random would have a small complexity, and things that are from nature or something like that we can see a lot of complexity would have this complexity measure higher. So paintings, ecological systems, and life, and the brain. So we could think about this kind of idea, for example, for networks, for graphs. We would have regular graphs and random graphs in these extremes of regular and random with small entropy and large entropy. But networks, complex networks like the networks of the brain networks, neuronal networks, or even cortical networks, the more complicated networks that we can find in nature would have these higher values of complexity. We could also think about complexity in time series. So if we measure any kind of quantity that is changing in time, so we have a dynamical system or something that we can measure, electrical activity, bold activity, whatever, we would also compare a very regular time series like this constant one and a random time series like completely random white noise with this, for example, an EEG data or alpha frequency that we measure during cognitive tasks. So we could be able to separate the complexity of this time series from the complexity of the very regular or very random time series. So thinking about that, it has been proposed a way of doing that if we use a symbolic information approach, and I will take a couple of minutes just to try to get you with this idea. So imagine if we have a time series here, and we decide to look for symbols in this time series. So we could think about 3 consecutive points, and then we would find, for example, that this point we have an increase, and after that we have a decrease. This would be correspond to this symbol here. But then after a while we have this increase symbol here that would correspond to the first symbol here. So for 3 consecutive points, we would have these 6 options and we can count how many times each symbol appears in our time series. So we can construct a PDF with a probability density function, for example, that show us how many times or the proportion of times that we find each one of the symbols that we decide to represent our time series. If we can do that, we can use 3 consecutive points or 4 or 5. We will have more symbols, but the idea is always the same. We can find this probability density function, and then this probability density function will represent our time series. So we can compare how complex this PDF, this probability function is. For example, comparing it with the very random case in which all symbols will have the same probability. So if we have a completely random time series, we would expect that there is no difference between this symbol or this symbol. So we would have a very flat distribution. Otherwise, if we have just an increase function, like an increase electrical signal, we would only find this first symbol, and our probability function would have this first symbol with all the probability, like 1, and all the others 0. So if we define things like that, we can calculate entropy in Shannon's-like idea. So the typical entropy with probability logarithm of the probability, summing all these possible symbols. And then we can calculate the entropy of our time series, but we can also calculate a disequilibrium, a Jensen channel divergence. So the distance of our probability function from the very boring probability function with everybody with the same probability. So when we define this disequilibrium in such a way that we can have the entropy using the probabilities and also a complexity using a normalized parameter, and comparing the entropy of our actual time series with the entropy of this boring time series, this very random time series, we can define this complexity in such a way that the random and the regular systems have a very low complexity, but intermediate time series like alpha frequency bands time series would have a complexity that is larger than the very simple ones. So I hope everybody's with me after that, but if it's not, just let's have an idea that we can do like this kind of such measure, like measuring complexity in complex systems. And now let's think about this idea of criticality in the brain, what that means, and then we can put the two ideas together. So let's think about criticality, which is a completely separate idea in research themes typically. And then in the last 20 years, I think there is this fancy idea among physicists working with neuroscience that we call the critical brain hypothesis, which means more or less that the brain would operate close to a phase transition. So physicists usually like phase transitions because they know how to explain, they know how to calculate things from systems that go into a phase transition. So these ideas become well spread in the community, and it's more or less like if the brain is suffering a transition or operate close to this phase transition, we could interpret this like if the brain was subcritical, so few neurons would make few others fire. So in such a way that if we stimulate our nose, for example, our olfactory system, if we try to smell a cup of coffee, we would really need to come very close to the cup of coffee to feel the smell, because we would need a lot of neurons to fire, to stimulate other neurons, and to perceive this activity. So this would be a subcritical system, and then we would need a lot of stimulus to perceive the smell of something. In the other side, if we have a brain that is supercritical and we feel the smell of the a cup of coffee in the morning, we would stay all day long remembering the smell of the coffee, because the activation of few neurons with molecules of coffee will activate a lot of other neurons, and will stay forever remembering the smell. So probably we are somewhere in between these two ideas, because we need an intermediate amount of stimulus of molecules to perceive the smell of everything. So this would be some kind of optimization process. And it makes sense, it's like a fancy idea, but then we have to verify, okay, if this is true, we have to really find signatures of criticality in the brain. So in 2003, Beggs and Plains shown that slice of rats can present neuronal avalanches, which are cascades of neurons firing that have a power law property, which means that the probability to find avalanches of a specific size obey a power law, which is this line in a log graphic. And this would be a signature of phase transition, a well-known signature in physical systems. So if the probability of have very large numbers of neurons spiking at a certain point in time is smaller than the probability to find a few neurons spike at a certain moment. And the coefficient of this line is well known, 1.5, and they verified that in these experiments. So people start to really say that this was experimental evidence for this possible phase transition. So the brain would be operating close to this critical point. And during the following years, other experiments have shown this kind of power law. And also, if you compare during these experiments the number of neurons that are active at a certain time window with the number of neurons that are active in a time window before, like comparing T plus TD and T, we have a ratio that the number of neurons on average that are activated is 1. So the number of neurons active is always close to 1, which is close to that critical point, saying that if this ratio was smaller than 1, so few neurons would be active every time window, so the activity will die. But if this ratio was so high, larger than 1, just few neurons would activate a lot of neurons, and we would have a very active brain, active region. So we would expect, if the brain was critical, we would expect to find this parameter close to 1, and they found that in these slides. So after that, with anesthetized rats and awake monkeys, so these power laws became quite appeared a lot, like quite ubiquitous in the literature. There are some kind of controversy if this is really the meaning of criticality, a phase transition. But recently, in 2019, this group from Recife, close to my city here in Brazil, they also show that besides these power laws, they could find other signatures of criticality in anesthetized rats. So they show that during anesthetized states, we can find many cortical states in the rat activity. So this activity could be quantified by this coefficient of variation. So when you look to the firing rate and calculate the average of the firing rate and the standard deviation, so you can quantify how the variability of spikings. And they found that during the anesthetized period, no task at all, the brain fluctuates. So the activity of the brain fluctuates, but this activity fluctuates around a critical state. So an intermediate state between very synchronized activity and very disordered activity would have this kind of power laws characterizing criticality. So they showed that if the brain has something to do with these phase transitions and is critical, it's not that it is critical all the time, but its activity fluctuates close to these critical conditions. So close to these regions in which we have these optimized capacity to perceive smell or to feel the coffee. So they have calculated this variability for critical states. They found this value, and this is quite very nice result because brings again this idea that okay, it's critical or not all the time, but it's close to criticality, and we can define the synchronized ordered cortical states or these desynchronized and disordered cortical states. But in the middle, in intermediate moments, we have these critical regions. How many time I still have?
We have we should be wrapping up. There should three about
okay.
3 minutes, for questions as well.
So just putting the two ideas together. Now, if we use the ideas of complexity measures in these critical states, so if we compare the very ordered and the very disordered states and calculate the complexity and the entropy. So we take this data from anesthetized rats and we use the entropy and the complexity that I mentioned in the beginning. And then we have, if we look, for example, for one rat, we have that the entropy decreases from disordered to ordered states as we expect. So if we have very disordered random things, entropy is 1. And if we have very ordered things, we have entropy close to 0. However, when we calculate the complexity, we find this kind of peak. So in intermediate states, like not very ordered, not very disordered, the complexity has a peak. And if we compare complexity and entropy in the same plane, we can still find this kind of peak. And this kind of peak coincides with the critical state that's found before with that CV that the group found. So if we compare not just one rat, but the 7 rats from their work, we still have this kind of behavior, so this kind of peak of complexity that is close to the critical states. So if we look to the variability of the activity, when the complexity is maximum, so we have a value of this variability that is very close, that is inside the range of the CV of critical states. So if we can define these critical states, close to these critical states, the complexity is maximum. And then we compare to a model, like a network model. All the details I will skip now. But if we compare to a cellular automaton model that we really know that the critical point is specifically happening for a specific parameter, we can still find this kind of peak behavior for complexity. So in the model, we really know where the critical point is, and we find that the complexity is maximized very close to this point. So this is the model in blue, and in gray, this is the data from the 7 rats. So even for anesthetized rats and the cellular automata model, we have these very disordered systems with random activity like high entropy and regular activity here. So both with small complexity, but the intermediate states have this larger complexity. So we could say that something is going on there, and this is expected for complex systems. So I would like to thank all the students involved, especially Nastara, who did the work, and my collaborators, Osvaldo Rosso, Mauro Copelli, which is the specialist in criticality, and Pedro Carelli, who is the experimentalist. And here we are in Brazil, in Maceió, so come to visit us after pandemic time. And that's all. Thank you.
Thank you so much. Okay, so we'll just have one question, and this is from John. Are there cases, like different brain regions or tasks, in which dynamics don't look critical, or do they look approximately critical in all cases that you've seen?
No, there are many cases that they could not find, like experimentalists could not find these power law conditions. So, and that's why there is still a debate that is it critical or not, or when is it critical? So during tasks, during anesthetized states, but it's typically people do not report the non-power law conditions, but yes.
Cool. Okay, great. Thank you so much.
Thank you.
So our next speaker is Carlos Ponce. Carlos is an assistant professor in the Department of Neuroscience at the Washington University in St. Louis. And today, Carlos will tell us about defining visual tuning via naturalistic image spaces.
All right. Hello, hello. Thank you very much for the invitation, and thank you all for being here. So yeah, first of all, let me just start by acknowledging the folks that did the work here in the lab. So this is a new lab, but it's going along pretty well. And today I'll be talking about work that was mostly done by Bin-Shu Wang. So in my lab, we study how the brain processes information about the visual world, specifically focusing on visual recognition right now. We study this in the macaque monkey using electrophysiology. And the overall goal that we have is that we want to understand how visual recognition happens in the real world, meaning in the context of images that you and I experience every day. They're complicated, they're textured, there's no clear segmentation, they're difficult to understand. And ideally, what we want to do is to be able to have a visually responsive neuron and present a random set of images or photographs, be able to interpret the responses and to predict what responses would be to new sets. And the fact is that we're not there yet. The field is not there yet. And part of the problem is that I think we've been dealing for the past few decades with some limitations, both technical and partially conceptual, that I think machine learning is helping us overcome. Specifically, we now have ability to parameterize images that are as complex as the natural world. And also, convolutional neural networks have really illustrated vividly and convincingly the idea that neurons really can serve as multidimensional filters. And this is an idea that's been in neuroscience for a long time. It's been illustrated well by folks like David Van Essen. And I think that this is now something that we can explore a little bit more. So to understand these ideas, let me go back to the beginning and just kind of remind ourselves how neuronal function is usually defined in visual neuroscience. This is a model, that experimental approach that we've taken from Kufler, Hubel, and Wiesel from many years ago. And basically the approach, you have an animal that you place in front of a screen while you introduce a microelectrode in the visual cortex, record action potentials or spikes, while stimuli are presented in the region of space to which the neuron responds. It's called a receptive field. And that allows you to get an image response function that generally looks like this. So how do we interpret that? There's been two major approaches to interpreting neuronal responses, and we can focus on this particular example of a neuron that is between V4 and Phototemporal Cortex in the macaque that was recorded about a month ago. And this neuron responded like this, a beautiful single unit, and it preferred these four images here on top. So one approach to describing what this neuron is encoding is to try and focus on semantic descriptions, which is great for communication between scientists, right? So we can talk about the fact that this neuron responded mostly to a face, so maybe it could be a face neuron. If most of the stimuli that it responded to were faces, we could have called it that, but really this is not that, because as you can see, it's also responding to rather abstract pictures like abstract art and an upside down house. And we can keep using words to describe this, but as Chris Olah brilliantly wrote in one of his articles, trying to describe the function of neurons using words can be a lossy operation. So we can move on and we can think of other ways in which it's been done. A very common approach is to use parameterized spaces, and this is well illustrated by things that were done, for example, by Hubel and Wiesel, where they found that neurons in primary visual cortex could respond favorably by eliciting more spikes to particular orientation. So here they have an axis that is well-defined orientation, and they can find locations within this space. that reflect the neuron's preference. And this actually has allowed folks like Christina and Rothkugel to very early on define probably the most useful tool in visual neuroscience, which is the tuning curve. This is a function basically tells us about the dependence of neuronal responses over a variable of interest, and is usually defined by at least three measures. It has a peak within this axis. It has a width that describes how responses fall off as you move away from that center, and it also tends to be pretty smooth, which is a useful feature to have for information theoretic purposes. So over the past 60 years, what's happened is that there's been many teams who come up with many different kinds of tuning curves, like contrast, orientation, curvature. And while all of these tuning curves do give us a very good functional profile of visual cortical neurons, really understanding the responses of neurons along these very basic axes don't really allow us to understand the responses in the natural world yet. So what can we do? And here, what we wanted to do is to focus again on the idea of neurons as multidimensional filters, right? And so what if, for example, if a neuron really is acting in a convolutional way, maybe what this is telling us is that the neuron is encoding some kind of motif that corresponds to some intersection, some combination of shapes, colors, and textures, and that all of these images just happen to have this feature embedded in them, and therefore the firing rate just basically tells us about a distance between the neuron's encoded template, encoded filter, and these particular images. So the overarching hypothesis, if we stick to that, is the idea that maybe the tuning curves that we've collected over the past, many decades really represent slices or samples of our larger tuning landscape. Here, the basic idea is that a neuron, anywhere in the visual cortex, for example, really operates over all images that fall on the retina. That's basically the domain. Anything that can be represented by its inputs, which is almost everything else. And so therefore, it's possible that all of the axes that we've created in the past just represent rather, specific samples of this larger context in which the neuron can respond. So that's what I really want to explore here, the idea that maybe there is something of a tuning landscape. And if so, there's two features that we can find. We can find a peak, at least one, which would correspond to a pattern learned from the world by the specific neuron. There could be multiple peaks. And we can also explore the idea of the tuning width, which should span larger image transformations over complex images. So that's what we're going to do. We're going to start with a tuning peak. So how do we identify a peak? Obviously, we want to do it over a naturalistic space. And at this point, the best way of doing this is a generative adversarial network. I've seen a lot of posters now that are using GANs in their projects, so I don't think I need to explain it too much. But basically, this is a deep network that has learned to take short inputs through a latent space, ranging from hundreds or thousands of elements, and then through a set of learned convolutional filters, is able to output an image. And they're usually very expressive. All of these images can really remind you of things in the natural world, and you can move smoothly from image to image. So what we did a couple of years ago, and this is work that was published from the Livingston and Kryman labs, is that we were able to take basically random input vectors, right, and propagate them into a GAN that is going to transform these vectors into images. We can show them to a monkey while we record responses from the neurons in the monkey's brain. Then we can feed these scores, these responses, along with the input vectors into a genetic algorithm that would then combine it and try to identify new candidate input vectors that would allow the neuron to increase its response. So this can happen closed loop within 1 or 2 minutes. And as a contrast to other approaches for activation maximization in the brain, we're not using data-fitted convolutional networks. This is just real-time interaction between the neuron and the genetic algorithm. So we started at first by focusing on Phototemporal Cortex, which contains neurons that are very complicated in the responses and can respond to things like faces or bodies and places. And as you can see in one classic experiment focusing in posterior Phototemporal Cortex, we started with images that look like this black and white textures, and we also used natural images that weren't changing as a function of reference. And what you can see is that this neuron began to change its firing rate as a function of generation, basically leading the images to change in such a way that it was eliciting a very high response that we can think as being analogous to equivalent visual's preferred orientation or to the center of a tuning curve. And so what's evolving? So what happens is, as I mentioned, we start with black and white textures, and then if we just average each one of these pictures per generation, you begin to see that indeed there appears to be a formation of a rather complex set of features that certainly are reminiscent of things in the real world. In a monkey's brain, you could see some kind of relevance. Here's some more examples that were collected in my lab, and as you can see, what these tend to be are diagnostic features that would be useful and diagnostic of things that the animal might care about, like faces, body parts, perineum, and even things that we can't quite understand. But we can interpret them, and we can do it in a couple of different ways. We can take, for example, a comparative approach, where if we have, once we've derived one of the specific synthetic images given a neuron, if you have a monkey that is nice and likes to work for a long time, and you can hold the neuron long enough, you can show that monkey another few hundred or thousands of images and identify the images in that set that also elicit good responses from the neuron. In this particular example, for example, it was pictures of monkeys. Another thing that we can do is that you can take these synthetic images and propagate them into a neural network along with hundreds of thousands of reference images, identify images that are closely matching per this neural network space, and then close the loop by showing them back to the neuron. And so that's what we did. And as you can see across these different monkeys, when neurons create some synthetic images, and they also respond really well to images that are near, according to this neural network space, compared to those that are far. And this is something that we've replicated many times past this particular study. So what this tells us, and right now I'm going to talk about these GAN-derived images obtained via this approach as prototypes, just for simplicity of communication here. But what this is telling us is that prototypes are probably marking a candidate peak in this overall tuning landscape over complex images. So now that allows us to ask the next question, which is, okay, so if this represents a peak, what does the tuning shape and width look like? And this is work that was done here by my grad student, Binshu Wang. He set out to define the shape and width of this tuning landscape around the prototype. And what he did is that he added an extra experiment. In addition to the evolution experiments that I just described, he added something called a manifold experiment. The evolution experiment takes us to some kind of peak. In this particular IT neuron, it led us to basically this shape that I like to call Mr. Duck Face, because it's got the two little eyes and this little kind of bill on it. And then what Binshu did is that he defined a two-dimensional hemisphere that is embedded in the original latent space of 4,096 different dimensions. And this particular hemisphere was defined along its latitude and longitude by the principal components that were orthogonal to the original trajectory that this neuron chose in this latent space. And that allowed the prototype to vary continuously and smoothly over the space. And as you can see by these firing rates that I'm superimposing here, it did look like that the shape of this tuning landscape near the peak was fairly bell-shaped and symmetric. So as you can see here by visual inspection, to formalize that intuition, what we decided to do was to fit a Kent function, a Kent distribution on this, which is basically a two-dimensional Gaussian sphere. It's used in geology. It comprises a lot of different parameters, but the ones that we really care about right now are kappa and beta. In those, if you take the ratio of those two variables and you happen to be in this regime, you would find that the shape of this is bimodal. If you're in this part of the ratio, it will be elongated, and if you're in this part of the ratio, it will be more symmetric. And as you can see, across all the experiments, most of them did seem to have a unimodal and bell-shaped form. So that leaves us the tuning width. How do we explore that? And obviously, we can describe it in the sense of this particular latent space, but we wanted to do something a little more ambitious. Because remember, the point is that these neurons are responding across all kinds of image types that you decide to choose to show them. So we wanted some space that would allow us to measure not just natural images or the GAN images, but also geometric, classic, powerful color stimuli, in all the way to contours and gabors. And so the way that we decided to do this was to use a perceptual similarity metric. And what these are metrics that basically are designed to emulate the subjective decisions or judgments that are made by folks when you're asking things like, all right, you have this picture here called this reference. Which one is more similar, patch 1 or patch 0? And so most humans would say that patch 1 is more similar, and these metrics are designed to try to replicate that effect. Right now, the best ones are based on neural networks. And so the way that they work is that you can input a couple of images, then the activations are weighed by specific, by vectors that have been derived from almost 500,000 human judgments. And that gives you a distance, that gives you a value between those two images. So here, the plan was to take, to measure the distance between the preferred stimulus in one particular space, like here in the manifold space, and all the other pictures in that space. And we can do that for natural images. We can do it for standard artificial images used in other studies. And that's what happened here. So for this particular neuron, a couple of things to illustrate. The first thing to show is that standard artificial images had a lower range of perceptual distances, right? And that makes sense. The largest distances were covered by natural images here in purple. As you can see, you can get all the way up to 0.75. The manifold images did cover the gap pretty well, didn't quite go as diverse as natural images, but as you can see, it does a much better job of sampling the space in between, which allows us to kind of more confidently, with more accuracy, define the tuning width of some of these neurons. Now, in this particular example, this neuron's prototype basically was as effective as the best natural image that we could find, which was this particular face. So not a duck, but a monkey. Nevertheless, across all experiments, we did find that all of these prototypes did elicit larger responses, which is basically a way of saying that the neuron was better at finding its own preferred features than we were at choosing for it. And this was true across different cortical areas. Nevertheless, if we estimated the tuning widths that these neurons exhibited by just doing a non-parametric AUC area under the curve, we did find that natural images elicited some of the wider responses. Nevertheless, we can be more confident because of the sampling using the GANs. So that's it. The basic conclusions here, the takeaway messages, is that we're pursuing the idea that these neurons really do have more than just tuning curves. They're really tuning landscapes. They're operating like multidimensional filters. We've shown you that these neurons do show large response ranges across image transformations that are comparable to those in the natural world, and that these landscapes are roughly symmetrical and unimodal. And that's it. If you're interested in learning more about our work, we also have a poster coming up at an international conference or learner representations and a couple of manuscripts in the way. So that's it. Thank you very much.
Great. Thank you so much for an excellent talk. So I have a question. You know, the particular choice of architecture, right, that you use for your generator will, of course, build in certain implicit biases into the network. And as you showed later, you know, the neurons actually found other images that were sort of better than the best, you know, synthesized ones. So I'm just wondering just your perspective on how to break through some of those barriers, or how those biases
absolutely.
are
We think that obviously the best way to, every generator has its own bias. There's more new ones that are more realistic, like Big GAN. We began to use those, and we actually collected a large data set where we actually pit different generators against each other in the same neuron, in the same session, to find out if things like photorealism, which is better in Big GAN and Style GAN, matters to the neuron. And the quick answer, without taking much time here, is that it doesn't seem to matter. We can evolve images that basically contain the same shapes. One is photorealistic and abstract. The other one, and for example, the idea of a pink eye that appears in both. One looks like a cartoon, one doesn't. The neuron gives you the exact same response. Which again tells you that these neurons are dealing with a much larger domain of images, transformations.
Yeah, very interesting. I think we have time for one more question. So someone asked, if you also take more top-down inputs into account, would you expect the maximally driving image to differ under settings with different
that's a great question, and that we started doing that. We basically now started recording from prefrontal cortex and visual cortex at the same time to figure out, first of all, what the output of the representation, so what areas that are more associative are doing with this information coming from visual cortex. And I guess it's too early to say what's going on. There are visual responses that we're able to examine, and we are about to put them together to answer that very question. So that's a great question.
Super cool. Awesome. Thank you so much.
Thank you very much.
All right. So we have our next speaker up. Tahra should be joining us on stage. There she is. Great. Hi. Okay, so up next we have Tahra Eissa. She's a research associate in applied math at the University of Colorado at Boulder. And today she'll tell us about strategic trade-offs between choice biases and accuracy for decisions based on asymmetric and rare evidence. All right, take it away. Thanks.
Thank you so much, Eva and Gabrielle, for setting this up. This is great. So I just want to start off by orienting us to what exactly I mean by asymmetric evidence and a lot of the issues that go along with the idea of asymmetric evidence. So let's take, for example, imagine that you're a physician and you want to diagnose a patient with a disease that has symptoms that are pretty sporadic. So for example, epilepsy. And you need to count whether or not someone has a seizure in this case, right? So seizures can be quite rare. You may or may not see one in a given time. So let's just assume that we have a symptoms log. So a week goes by, your patient returns, and they haven't had any symptoms that you can log. If we were to stop looking at the evidence at this point, we may be swayed that our patient doesn't have epilepsy, right? Which may not be representative of the truth. But if we go for another week, now we see we have one day where there's an episode where they have this seizure. And in this case, we see now that, oh, well, what I mean by asymmetric evidence is in our decision, this one day should weigh more heavily in my choice and my diagnosis than all of those other days where we don't see anything. So when we say asymmetric evidence, what we're really talking about in this case is asymmetric evidence weights. And as we see in this example, if we have limited evidence, it can be easy to be swayed against the correct answer, right? Or the right choice. So we decided to start off by asking, okay, how would an ideal observer approach this type of a question? And so in order to answer this question, we're going to consider a task that's just a very basic jar discrimination task. So in this task, our observer is going to see two jars, and they're going to have varying ratios of red and blue balls. The observer knows that these balls are, the jars are equally likely. And they're shown a sample of balls drawn with replacement. And then the observer must decide which of the two jars they think is the source. So in this case, what we've done to make the symmetry different is to say that our asymmetric jars are going to have one color, so here it's red, that is going to be rare in both jars. In the other case, we have symmetric jars where the ratio of red and blue balls are reciprocals. So there's three components that our ideal observer is going to consider then. The first bit is the prior, which is going to say, okay, going in, I know each jar is equally likely. Then the second component is going to be our evidence weights. So because the ideal observer knows the exact ratios in our pair of jars, we can do this based off of the ball likelihoods and just ask, well, what's the probability that a particular observation came from either of my two jars? So then we're going to add these things together by comparing our decision rules. So we're going to use a log likelihood ratio in which we say, of all my observations, I'm going to look at each one individually, and I'm going to compare the log of that ball likelihood evidence weight. And at the end of this, that's going to shift my belief. So if my belief, Zn, is positive, then I'm going to respond in favor of the high jar. And if it is negative, I'm going to respond in favor of the low jar. So if we just look at the example that we have up here with all of these blue and red balls, we can see what we really mean about this asymmetric evidence. So over the course of time, our ideal observer is going to shift their belief with blue balls encouraging me to choose the low jar, and a red ball shifting me towards belief in the higher jar. And we can see that the green trace, our belief is shifted more when we see a red ball than in the orange trace. So keeping this in mind, then we can consider, well, what happens over many, many, many trials? And what we'd expect is that the responses in favor of the high and low jar would match this prior. So we'd have about half in favor of each. But what ends up happening, because we're only considering limited evidence, so in this case, we consider what happens if we show 2, 5, or 10 balls, is that we actually end up with a choice bias. So the ideal observer actually has a preference for responding in favor of the low jar more than the prior would suggest. And so what this really comes down to, and we can imagine that makes sense, is those red balls are rare. And so the chances of observing them are much lower in a limited evidence case. So we noticed that this choice bias existed. And the obvious next question is, well, how do humans deal with the same task? Do they also show a choice bias, or do they recognize that they are going against the prior and shift some part of their belief, right, or their strategy? So what we did was we gave human subjects the exact same task, and we varied the difficulty as well as the symmetry. And what we found is, just as we saw with the model, our human subjects also showed a choice bias, specifically with respect to those asymmetric jars, and not with respect to the difficulty. And similarly, in line with what we saw with the ideal observer, we saw that this choice bias was most extreme when you had limited evidence. So when our subjects were only shown 2 or 5 balls, they were going to show a stronger choice bias. So qualitatively, just looking from the get-go, it seems like our human subjects are using a really similar strategy to the ideal observer. They're showing the same biases that we would have expected. And so the next question is, well, let's look at this at a more individual level. Are these biases really the same? And the way that we're going to do this is by considering what we call these response sequences. So at any given block of trials, what we can do is compare what I respond on one particular trial to the next one in sequence, right? And so here I would go from high to low, which would be considered an alternation. Then I would go low, low, a repetition. And what this allows us to do is, by comparing the fraction of times that we go from low to high or high to low, we can actually differentiate between different forms of biases. So what we'd expect the ideal observer to have is what we said was a low jar bias, where they're often going to switch after a high jar choice back to low, but they're not often going to switch from low to high. In contrast, previous work has shown that humans also show sequential biases. So maybe that there are non-normative effects like a repetition bias, where you prefer to respond with the same response over and over again. So plotting our subjects across all of the blocks in this space, we see that, as we expected, most of our subjects in those asymmetric cases, so the green dots, are shifted towards that low jar bias. So they are showing that same ideal observer bias that we expected. But because we know that the ideal observer also has this bias, we're going to take this one step further and ask, how does each individual compare between their choices and an ideal observer model that has been given the exact same set of samples? And so looking at this deviation, we actually find something really surprising, which is that we would have expected if subjects were doing exactly what the ideal observer is doing, they would end up right in the center here. But instead, there's a substantial number of subjects that are actually shifted up. And this is what we're going to say is an excess low jar bias. So they're actually showing that they have an increased bias beyond what the ideal observer would have predicted. And so this goes against what we initially thought may have happened, which was that subjects would be uncomfortable by this bias and maybe adjust in the other direction. So now we're asking the question, what strategy could be causing this? Where is this bias coming from, this excess bias? And to answer that, we're going to look at two forms of model classes. So the first one are these Bayesian-based models. And so this noisy Bayesian model is going to be kind of our base model for comparison. And in this particular form, we have a Bayesian update with some noise, and then we have a free parameter rho, which is going to measure and fit what that rare ball weight is, that asymmetric weight we were talking about. And then just very briefly, the other forms of this model class is going to be a noisy Bayesian where we set that rare ball weight to what the ideal observer has. We have one where we have a prior added in, so maybe there is actually a biased prior to begin with, or maybe our subjects like to have just a particular amount of evidence. So this is what we're going to call a windowing model. Then our other class of models, we have our heuristic models. So really we have two flavors of our heuristic models. One are the rare ball models, where we're going to say if I see any rare balls in my trial, then with some probability p rare, I'm going to respond in favor of the high jar. And then if I see no rare ball or no rare balls, I'm going to respond in favor of the low jar. And then we have one version where we say, okay, I'm going to have a rare ball model and think about what I did on the last trial. And finally, just a guess model. I don't even care what's on the screen. So we compare these using Bayes factors, where we can say, well, what's the probability that a response came from some alternative model as compared to that base model, our noisy Bayesian model? And what we found was that our subjects predominantly used a Bayesian-based approach. But, and this is, I guess, key to this whole concept, not necessarily optimal. And so this brings us back to the question of, well, what about those subjects with the enhanced choice bias? So if we go back to our bias plots, and now we color code our subjects based on their best fit model, we see that most of the subjects that were up in that top left quadrant are purple. So they're Bayesian based. And really, they come from two of our particular models. The first is the noisy Bayesian model, where we allowed it to fit for the rare ball weight. And we see that the subjects that have this low jar excess also underweight their rare ball. So here, these black lines are where the ideal observer would set the weights. And we see that all of these subjects are underweighing in comparison. The other set of subjects that show this were using a prior Bayesian approach. And so they actually had a biased prior to begin with. So this is really interesting in terms of explaining individual variability. But we can take this one step further and actually say, well, can we understand anything else about these model classes themselves? So if we look at how our subjects compare to their ideal observer, both heuristic models and suboptimal Bayesian models are going to all have an accuracy decrease. So they're going to lower their accuracy compared to the ideal observer. But those that were using a Bayesian-based model also showed a really strategic trade-off where their bias increased as their accuracy decreased. And so the question was now, well, is this just representative of our particular set of subjects, or is this something we can say more substantially about these model classes? And so we generated synthetic data that from these same model classes, and we can see that for these asymmetric cases, this strategic trade-off really is specific to our Bayesian-based models. And so this goes beyond just individual variability. So in summary, I just want to reiterate this idea that we've seen that both an ideal observer and a human subject shows a choice bias when shown asymmetric evidence. Our human subjects tended to use a Bayesian-based approach. And as we decrease our accuracy from the ideal observer, Bayesian approaches are the ones that show this increased choice bias. So lastly, I would like to acknowledge my mentors, Zach Kilpatrick at the University of Colorado Boulder, Kasia Yoshik at University of Houston, and Josh Gold at the University of Pennsylvania for all of their help with this.
Thank you, Tahra. We have a question in the chat. It's a clarification question. What are the differences? What are the differences in predictions of these models? Bayesian, different versions, and non-Bayesian ones.
The predictions in terms of accuracy, or in terms of what exactly? I guess.
I'm not sure if, but can you respond to both of those actually?
So in terms of accuracy, we would expect that accuracy is going to decrease for either type of model if you're not using sort of these optimal parameters. But I think what's really key in this particular situation is that we're saying that when you have your heuristic models, you're decreasing your accuracy, but you don't have any of these sorts of biases, right? If anything, you're actually almost going the other way. You're sort of got this like anti-bias thing going on in some cases. Whereas when you decrease your accuracy and you're still trying to fit that, it's really hard to fit a Bayesian approach in your brain, right? That's a lot of effort. And so to do it well takes like a lot of finesse. And so what this is really saying is, hey, if you get this wrong, not only does your accuracy decrease, but you actually have this bias increasing, and there's this really impressive trade-off that you see. I'm not sure if that answered the question.
There is another question that has popped up. Would you predict these effects to become more extreme with more asymmetric choices as humans deal even more poorly with extreme chances?
Yes. So actually, there's a whole body of literature that looks at what happens when humans are faced with rare events and estimating them. And actually, it seems to go both ways depending on how you structure the task itself. And so you can actually have subjects that overestimate and underestimate depending on how you pose the task, which is kind of cool.
Excellent. I'll ask one more question from the chat. Are there other contexts in which you can explain deviations from ideal Bayesian decision strategy as some kind of trade-off?
That is an excellent question and something that we're thinking about right now. So get back to me in a little while and hopefully I'll have more answers.
Excellent. Thank you so much for that talk, Tahra.
Thank you for having me.
Thank you. Our next speaker should be coming up right now. Jay Gill. Hello.
Hello.
Jay is a PhD / MD student at the University of California, Los Angeles in the Cal Lab. And today, Jay will share their work on oscillatory encoding of emotional valence in humans with and without anxiety-related disorders.
All right. Thank you for the introduction, Gabrielle, and hello, everyone. Thank you for coming. So as was just mentioned, my name is Jay, and I am at UCLA working with Drs. Nanthiya Suthana and Jonathan Cao, and I'll be presenting some of our early evidence trying to see how humans with and without anxiety disorders encode valence using oscillations. And so while I was making this presentation, I thought of this pretty common saying, fool me once, shame on you, fool me twice, shame on me. And I decided to use it because I think it does a really good job at motivating how important it is for anyone that cares about their survival to store memories of aversive outcomes and use them in the future to guide actions to avoid them. And so at all stages of the evolutionary tree, we can observe how critical threat detection and response systems are for survival. So in the case of the mouse, we can think of the importance of remembering the scent of a predator like a cat, and how when that's encountered, it might want to hide or freeze. And jumping up the evolutionary ladder to humans, we can see how these systems might help us remember to not touch a hot stove. And so although these systems are absolutely essential, when they are overloaded, they can result in pathology. So in the case of PTSD, this may be an inability to escape a trauma that was once encountered, or in the case of anxiety, it could be persistent worry about a potential distant aversive experience. And so these disorders, which can be grouped as anxiety-related collectively, affect about 40 million or 20% of US adults each year. And despite their vast prevalence, only about 50% of patients experience some benefit for currently available therapeutics. And so this really motivates why it's so critical for us to really dissect these circuits so that we can really lead to the next generation of targeted and more effective therapies. And so before I introduce our methods, I wanted to provide a brief overview of neural circuits implicating and mediating threat, processing, and response. And the story typically begins with the amygdala, which is an almond-shaped structure that's a collection of nuclei in the medial temporal lobe. And so this structure receives a swath of sensory input and is known to maintain the relationship between a stimulus and its outcome. So if we think about rodent fear conditioning experiments, it's the place where rodents store the memory that a certain tone is paired with a shock. And so that when that tone is encountered in the future, rodents can exhibit a fear response. And so the amygdala doesn't act in isolation. Its activity is regulated by the prefrontal cortex as well, which can inhibit or enhance expression of a fear response. And lastly, the hippocampus also plays a role and delivers context to the circuit, so it can allow animals to realize that stimulus outcome associations in one environment may not generalize to another. And so through decades of research in rodents, it's been identified that these structures implement their respective roles and communicate with one another via changes in neuronal oscillations. And these just characterize the frequency at which populations of neurons change their activity. And much of rodent research has focused on activity in the theta range, which is about 4 to 7 hertz, and in the gamma range. And at a very simplistic view, it's been found in rodents that threat is associated with theta and fast gamma is associated with safety. And although we've been able to pretty much characterize these circuits and their oscillations fairly well in rodents, what's going on in humans is particularly unknown. And so that's this wide, really interesting area of research that we're hoping to contribute to. And so to provide insight into this unknown, we showed 8 human participants images that were either negative, positive, or neutral. And during image presentation, we recorded activity from the amygdala and the anterior hippocampus. And to give context for how we're able to conduct this type of investigation, we work with patients receiving neurosurgical care in 2 scenarios. The first is while patients are undergoing localization of their seizure foci while they have electrodes implanted throughout the brain, so we can come over to the hospital and perform tasks with them. The second is patients with chronic implants called a responsive neurostimulator that's constantly recording activity from the brain and listening for detection of maladaptive activity, such as that is associated with the seizure. And so through both of these advancements, we're able to record LFP and neuronal oscillations from deep brain structures in humans. And so through this setup, we hope to answer the following questions. So 1, does frequency band power reflect emotional valence? And 2, because we have participants, 3 of them, who have an anxiety-related disorder like PTSD or generalized anxiety disorder, we wanted to know if this oscillatory encoding of valence was dependent on anxiety status. And so to quantify the relationship between valence or emotional charge and neural activity, we looked at the prevalence of different network oscillations during the viewing of different image types. So here I'm showing an example time frequency power spectrogram from a participant's right amygdala. And so the x-axis is time, the y-axis is frequency, and the color is the normalized power or strength of a oscillation in that frequency. And this dotted line is denoting the onset of a negative image. And so because we were interested in seeing how spectral power changes in response to images of different valence, we examined the total theta, alpha, and gamma power in the right and left amygdala and hippocampus during negative, positive, and neutral images. And so when we're just looking at these average spectrograms for negative, positive, and neutral images, qualitatively, we can observe that there are differences between these conditions. And so to statistically evaluate this, we calculated the total frequency band power in these windows for the specified time duration. And when we did this across our electrodes and the different frequency bands, we found significant differences in right amygdala theta and also right amygdala gamma power. Specifically, we found that across subjects, where each subject in these plots is one of these dots, we found that during negative images, which is this red bar, there's higher theta power compared to positive images, which is this blue bar. And we also found that in the right amygdala as well, during negative images, there's greater gamma power compared to during neutral images. And so interestingly, when we broke up this gamma effect and stratified it by anxiety type, we found that only in the individuals without anxiety, there was a significant increase in gamma power during negative images compared to neutral images. And so though the trend is also present in the anxiety group, this lack of a robust differentiation between negative and neutral stimuli may suggest that individuals with anxiety have altered oscillatory encoding of valence. And so these results were really interesting because they corresponded with previous research. So our first result, that theta power attacks negatively charged stimuli, has been pretty well documented in rodents, namely during fear conditioning tasks, where this red curve is a tone that was paired with a shock, and this blue curve is a tone that was not. We see that during the shock paired tone or fearful tone, there is a selective increase in theta power. And so this suggests that there is sort of this cross-species utilization of theta power to tag aversive or unpleasant experiences. And our gamma result also corresponds with previous research. Namely, we found that increases in gamma power follow negative images selectively in subjects without anxiety. And another study by Zhang et al. That was conducted in humans with epilepsy, they found that during aversive or fearful phases, there was an increase in gamma power compared to when neutral scenes were shown to subjects. And this is this green curve here. And so though we found this very interesting, we were curious about how changes within different frequency bands across the hippocampus and the amygdala may work together to tag stimuli in the environment as negative, positive, or neutral. And to address this unknown, we use a pretty basic machine learning tool, multinomial logistic regression. And so we started by passing in 12 features, which included the total theta, alpha, and gamma power during the first second of image presentation from the left and right amygdala and the left and right hippocampus. And so we trained the model to classify whether an image was either negative, positive, or neutral. And so we found that when training a model to classify these different image types, it learns to predict image types significantly above chance at around 43%. And because we were also interested in seeing how the inclusion of data from anxiety subjects affects these results, we trained another model only using data from individuals without anxiety, and that's this green bar here. And we found that the model actually improves its performance and obtains an accuracy of about 47%. And so we next were really interested in understanding which regional frequency band changes were most important in classification. And so to do this, we examined model weights. And so when doing so, it was pretty clear that the model coefficients converge to different values during classification of negative images, positive images, and neutral images. So as an example, if we look at right VLA theta, we see that when the model is differentiating is attempting to differentiate negative images from the rest, it has a very high value for the right amygdala theta power coefficient. However, when it's differentiating positive from the rest of these two other classes, it has a very low value. And so this presumably aids the model in discriminating between negative and positive trials. And so to interrogate which of these weights were most useful in classification, we plotted the distribution of model weights across cross-validations and looked at which features had values that were below the 2nd percentile and above the 98th percentile, under the understanding that these values would play the largest role in determining whether a trial was negative, positive, or neutral. And so this approach identified 5 features: right amygdala and left amygdala theta power, right hippocampal theta, right amygdala gamma, and left hippocampal alpha. And so when looking at the weights for these features under the different classification problems, so negative versus rest, neutral versus rest, and positive versus rest, it's clear that the value discriminates between the different trial types, really emphasizing their role in supporting model performance. And so to further quantify how these features affect model performance, we looked at accuracy when only these top 5 features were used for classification. And we found both in the models that included here and excluded here data from anxiety participants, the model with only the top 5 features is sufficient for the classification problem. We then wanted to see what would happen when we removed these features, and we found that, interestingly, the model that doesn't see data from anxiety participants is not better off, but is less affected by exclusion of these top 5 features. And from this, it kind of let us know that perhaps individuals without anxiety have more widespread encoding of valence. So there are other frequency band power changes that are supporting the discrimination between negative and positive and neutral trials. And so lastly, we wanted to characterize the relative importance of each top 5 feature in the 2 different models. So again, the model that saw data from anxiety participants was in purple, and the model that didn't was in green. And here we found that in addition to the less robust widespread encoding of valence, in the model that was trained on data with anxiety participants included, there was an enhanced utilization of theta power to discriminate between image types. In particular, we see that when theta power is removed here, there is the most drastic reduction in model accuracy. And so this suggests that individuals with anxiety may be sort of relying on theta to discriminate between these different image types. And so that is our main results. So I just want to bring us to our conclusions. And so in summary, we found that during unpleasant stimuli, amygdala theta power is increased, and as well as gamma power. And so we found that negative related gamma changes are only significant in individuals without anxiety. And lastly, using that multinomial logistic regression, we found that there is less robust encoding of emotional valence in individuals with anxiety. And that there is an enhanced dependence on theta power. And so it's interesting because theta has been implicated in imminent threat detection in rodents, so it might suggest that individuals with anxiety disorders may be relying on this system that is sort of known to cause individuals to react, and that might be related to their disorder, but of course, that requires much further investigation. And so with that, I would like to thank you all for joining me today and the rest of us at this panel. And I want to thank my advisors, Dr. Nanthia Sudhana and Dr. Jonathan Cao, as well as the members of the Sudhana and Cao labs. And then also I want to give a huge thank you to Dr. Jean-Philippe Langevin, who is the neurosurgeon that we work with at the VA in the MD PhD program that I am a part of. Thank you.
Excellent. Thank you so much. Okay, so I have a question. I wanted, I know that you were adding in, coming from amygdala and then also adding in hippocampus. And so I was wondering if you've looked at interactions or coherence or or how these two different brain regions might be coupled in some way. And
absolutely,
and using
that was one of our first
study.
questions. And so we looked at phase amplitude coupling and then also coherence between both within and also between the amygdala and the hippocampus. And so we didn't find anything significant. We think that it might be due to the short period that the image is shown. So it's only shown for about 4 seconds, and typically those measures are used with data that's about 15 or 20 seconds for a reliable measurement. And so we think that might have something to do with it. And so in addition to this, we also do fear conditioning, which is over a much longer period that we can look at. And so we're hoping to see in the future if we see something there.
Interesting. Okay, we have a few questions coming in now. So Eric asked, can you talk more about differences between the hemispheres?
Yeah, that was also really interesting to us. So the effects that we presented today were largely coming from the right hemisphere. And so there's been some evidence of differential activation of hemispheres in fMRI and single unit experiments, namely some of the data from the Phelps group during fear conditioning has shown greater activation of the amygdala and prefrontal cortex on the right side for fearful stimuli compared to the left side. And then also in single unit, there has been differentiation of like the decodability of negative versus positive stimuli. And so we're seeing some hemispheric differences here, but we have a limited data set, so it could be a difference, just like a low trial count, and it might be a very small effect. So I don't want to put too much weight on that claim and say that there's a differential encoding of valence on the left and right hemispheres, but from what we're seeing currently, it's stronger in the right side.
Interesting. Okay, so we have another question from Holly. Have you looked into people with other neuropsychiatric comorbidities, like both PTSD and autism, or just other groups?
Yeah, so interestingly, the patients that we work with have like a very interesting history. And so it comes with a variety of mood disorders in addition to anxiety disorders. And so primarily, since we're at the VA, we focused on anxiety-related disorders just because PTSD is fairly prevalent. And also individuals with epilepsy have a higher prevalence of anxiety that's normally related to their seizure. So we focus there. But in the future, we're definitely hoping to expand it just because this patient population is so diverse and really interesting. So it's very cool to get the opportunity to work with them.
Okay, I think we have time for another question.
Okay.
You gave us a lot of time to work. Could the higher performance of MLR, so the logistic regression trained on non-anxiety subjects, be due to higher variability in data for the anxiety subjects?
Yeah, so that is our idea. We think that higher variability, or just we think that higher variability means that there's just less characteristic encoding of valence, and that might be related to their behavioral dysfunction. That's sort of a symptom of their disorder. But yeah, we want to get more individuals in both of those groups to see consistently if there is a particular deficit in the group and not just enhanced variation.
Okay, I think we have time for one more question. I'm going to ask a question now because this is something I was thinking about. Okay. So, you're forming this logistic regression. You're splitting these different states, right? Negative or positive or neutral into discrete classes.
Yeah.
But yeah, I'm just wondering kind of what your thoughts are on encoding this in some continuous way, or having some way of not discretizing perhaps emotional state in some sense.
Yeah, that's a very interesting point. So one of the motivations for discretizing it was because, within the negative category, we're just looking at images. So they are unpleasant. So one example is like a war scene or an emaciated child. So, the way in which, or the degree to which both of those is negative is different based on the person and based on an individual's experiences. It's quite personal. And so we thought by placing things into these larger categories, we might be able to get out a more robust result. But I agree, you know, doing it along this continuum is something that's very, very interesting. And so we're starting with the rigid categories. And I think in the future with more data, we want to definitely open it up to something that's more along a continuum.
Excellent. All right. Thank you so much for sharing your work with us today.
Thanks for having me. Thank you.
All right. And so we'll move on to our last speaker of the session. So we're joined by Shri Devi Sharma. She is an associate professor in biomedical engineering and also the vice dean for graduate education at the Johns Hopkins University. And Shri will tell us about when primary motor cortex drives premotor activity during movement and using dynamical systems modeling to challenge conventional perspectives.
Thank you, Ava. That's a long title. I know. Well, thank you so much, Gabrielle and Ava, for inviting me. It's so exciting to see these young, bright scientists and the colorful presentations and speakers. I have not come to Cosyne in a long time, so it's wonderful to see this. So thanks for putting this session together. So, I'm Sri Sharma. I have this little slide here with the math because I am purely computational, but that's not the kind of math we're going to talk about today. So what I'd like to talk about was work done by my PhD students about to defend in a week or two. Her name is Reina Deleo. She's a PhD student in the neuroscience department at Johns Hopkins. So today we're going to ask a very general question that I think most neuroscientists are very interested in, which is when you have multiple regions in the brain, how do they interact to govern behavior? It's a very high level question, right? But if you think about the challenges a neuroscientist face, even if they're really focused on a specific system in the brain, they train multiple subjects to execute a very structured task, and they record from very targeted regions in the brain. And they try to replicate this across subjects so that they can, try to come up with some general principles of how those regions govern behavior. But the problem is when you do this and you are recording from multiple regions and you're trying to see what their distinctive roles are, well, first of all, they all might co-activate during the entire behavior, okay? And worse, if you start measuring from different brains, even if it's trying to target the same areas, the neural activity across the subjects may not look the same, even if their behaviors look nearly identical. So these are the challenges we face when trying to answer this general question. So I'm going to take a crack at it, where we're going to focus on an example, a case study of the motor control system. So we're going to be interested in how premotor cortex and primary motor cortex, M1, interact to drive movement. So for those of you who are sort of new to sensory motor control, this is a very simplistic diagram. The idea is, let's say you want to reach for a glass of water. You have a goal. This goal, the intent is sort of processed in goal areas, parietal lobes. That information ultimately gets sent to motor cortices. And the conventional thinking is premotor plans, sends that plan to the primary motor cortex, and both actually project signals down to the spinal cord to innervate the muscles that allow you to move. But the conventional thinking has been predominantly that this is kind of a feed-forward pathway. Of course, there's proprioceptive visual feedback, but it's PM that then talks to M1, and M1 sends the commands. This is sort of a conventional viewpoint, but it's being challenged. But I want to discuss it a little bit. So why is this sort of the conventional way? It's even taught in our introductory to neuroscience, just conceptually. Well, maybe because premotor cortex has an interesting anatomical location. It resides right between decision-making areas and primary motor cortex. So it's kind of in an ideal location to kind of plan, okay? There have been a bunch of tasks over 2 decades ago that sort of impose a delay period between a stimulus and the actual movement onset. They kind of force this delay, a planning period, and then they record from premotor cortex and primary motor cortex and start seeing that, oh, okay, premotor areas are activating before the motor areas. Again, sort of reassuring this conventional view. Motor cortex, primary motor cortex, does have more connections to the cortical spinal tract through to down to the spinal cord. Even though premotor does as well, primary seems to have more. So it seems like it's more of a good final command output station is primary. And pre was labeled pre because phylogenetically it's older than primary motor cortex. But there are good reasons to challenge sort of this conventional viewpoint, okay? First of all, natural movements don't involve delayed planning periods. We can't just stop, think, plan, move. That can happen almost instantaneous, simultaneous. And of course, these are pros, and they've learned these types of movements. It's muscle memory, as you wish. But still, naturally, we don't have these imposed delay periods where we can plan. Another reason to potentially challenge this conventional view is that there's reciprocal connections between all these areas involved in motor control. I show some more than premotor and motor. I show supplementary motor here as well. But if you focus on premotor and PMV, premotor ventral and motor cortex there, they actually have reciprocal connections. It's not just PM projecting to M1, M1 going down the spinal cord, M1 projects back. So why can't it potentially drive PM? Why does it have to be PM feed forwarding to M1? Can't it feed right back? And finally, I hope is that we're going to leverage dynamical systems modeling to try to address these challenges and also challenge the conventional view. Okay, so that's where we're going. And our case study, we're going to use an experiment by Dr. Mark Schieber, the University of Rochester. So this is his experiment. So he trained 2 non-human primates to execute a reach, grasp, manipulate task. And there were 4 different trial types in this task. A cylinder, so the monkey had to go to the cylinder and pull it. That was the manipulation. Or a button, press. The monkey moves in there and presses it with his finger. Or mallet, reaches for the mallet and kind of pulls the mallet. Or a sphere object where they have to reach and turn the sphere. Okay, so while the monkeys were executing that task, let's see here. Oh, I need to go. Oh, sorry. I don't know how to fade forward anymore. Why is it? I apologize here. Let's see if I can do something here. There we go. Okay, so this is what a typical trial looks like. Okay, so initially there's an LED. The monkey holds a home center object, and then an LED cue turns on. Once the LED cue turns on, it's going to point to one of those 4 objects. The monkey moves to that object, holds it, manipulates it, and stops. Okay, and at the same time, in these 2 primates, Dr. Schieber recorded from areas both in the primary motor cortex in the blue and the premotor cortex in green using floating microarrays. He also captured on the hand, he put 30 sensors on the hand to be able to capture XYZ kinematics, okay, sort of path in 3D space. Okay, now with that data, of course, we saw all the challenges that I mentioned above. Both premotor and motor areas co-activate. The 2 monkeys, I'm going to show you a case here on the left, a video, where the monkeys are executing the cylinder task. And if you look at the top is the behavior, okay, I'm going to play it again. The bottom is sort of overall activity in premotor and motor. So blue is overall activation in motor cortex. Green is overall activation in premotor cortex, okay, across many neurons. But I want to call to your attention is if you look at the behavior, I'm running this video again, the behaviors look very similar across the monkeys. The neural activity, okay, you kind of see this shape pattern, but in one case, it looks like motor cortex exceeds activation over premotor. In the other case, it's not totally identical. Similar situation here. Again, this is a gross summary across all the neurons, but here, looking at the sphere, again, the behaviors look similar, but now here the neural activity looks quite different across the subjects, monkey X and monkey Y. And in particular, in monkey X, premotor activity is much higher than motor activity throughout the sphere, and it's not so distinguishable in monkey Y. So it begs the question, okay, how can we learn about how these cortical regions drive behaviors from multi-subject data? How do we get at the consistency question? We took all our data and we asked, okay, what we would want to happen is the more the behaviors of the two monkeys deviate, so that's the x-axis here, kinematic difference, the more the different the kinematics, we would expect the more different their neural activities in both of these areas. And we would expect that there be a relationship there. And the closer the behaviors, the closer the neural activity. And that's not really what you see. In fact, it's kind of vertical here if you kind of look at the thing. So no clear relationship. So how do we make any conclusions about what's going on across the monkeys? Well, one of our first hypotheses, well, let's do what many people have already proposed to do. We need to have the neural basis being the same across the two monkeys. So monkey X might represent information in its brain using one basis, say a standard Euclidean basis, and another might choose to represent the same information in a different basis. So let's just do a simple linear transformation, a rotation operator, and basically project monkey Y's neural activity into monkey X's basis. So this just happens through a simple linear transformation unity gain. So we're just rotating the data and then say, okay, now are they more similar? The more similar the behavior, the more activity. Well, first, we want to see how well the reconciliation was. So what you're going to see here, this is firing rates. Each of these plots show firing rates of populations of neurons in the top row, M1, primary motor cortex. The bottom row is premotor. And let's focus on this top left plot here. This is one cluster firing activity across a trial, Q, movement onset, and switch close. So they finished the manipulation. The red is monkey Y, the blue is monkey X in their own bases. And you can see this cluster, they don't look anything alike, the firing profiles. But once I transform the basis of monkey Y into monkey X, they reconcile a bit. That's the purple. So the purple is the transformed firing activity. And you can see across all these various clusters, even in pre-M, we are able to reconcile the firing rates to some degree. Okay, they're not identical, but they're much closer. And in fact, if we look at the response errors between the firing rate profiles of all the clusters across all the different tasks here, we see that after transformation, we reduce the overall errors between the firing activity in every single population. We talk about populations of neurons because what we did a priori, we have 100 neurons, say in premotor, 100 neurons in motor, and we cluster them within structure in such a way that a cluster of neurons is a set of neurons that fire together across all four task types. Okay, so they have similar firing rate profiles, and that's how we get populations. But that reconciling of basis didn't help us get back that relationship. So remember, if you look at the right, this was the plot I showed you earlier. I want to see the more the behaviors deviate, do the neural activities deviate? Is there some nice relationship? We didn't see it before, and guess what? When you transform the basis, you still don't see it. That may not be so surprising, but we wanted to give it a shot. So what is it that led us to our second hypothesis, which is maybe it's not about the neural activity, the observed responses that have a linear relationship. Maybe it's something about the internal dynamics, what's going on in terms of who's driving who at any given moment in time, cortical drivers, which I'm going to define in a second. Maybe cortical drivers are more similar when the behaviors are similar. And it's not something I can just measure with electrodes or wires. So let's talk about that. To get at the neurocortical drivers, we're going to build dynamical systems models. So you might have seen this before, introduced many years ago from the Shenoy, Churchland group, and so forth. And the idea is following. I'm going to represent neuronal firing rates or firing rates of populations in a neural state space, which you see on your left. And this is an example of 3 neurons, so it's a 3-dimensional neural state space. And trajectories in the neural state space are going to govern behaviors or the physical kinematic space. So the idea here is, let's see if I can, there we go. I'm going to say x of t is a 3-dimensional vector where this is the firing rate of each individual neuron, x1 through x3. And I started in some initial baseline firing rate. Okay, I get a cue. That's like kicking the dynamical system with an input. That's just a LED cue that says this is where you need to go. And once the monkey reacts to it and initiates a movement onset, that generates a trajectory in the physical kinematic space. So X will lead to a trajectory in the neural state space, will lead to a trajectory in the physical space. And we model those dynamics, you might have seen this before, in linear time invariant dynamics, okay? So you have a state evolution equation, which shows how the neural firing rates evolve over time. And then the bottom equation is what's called an instantaneous output. It's how the kinematics relate to the firing, the neural state. And this is just a linear, simple linear relationship. It's a simple model. So you wonder, why do we start simple? Because that's what we want to start with. Because if it does the job, we'll stop there, okay? So how do we build this model from data? We're going to assume the LED cue, that flash of light, and the movement onset are like exogenous inputs to the dynamical system. So those are my U of T's, okay? And my outputs, my neural states are the firing rates of the populations of neurons. So I have a bunch of populations in premotor, populations in motor, 6 and 6 in this case, and they interact dynamically through those equations, okay? Just a schematic of this. When we estimate those parameters from training data, model overlaid with data, models are the colors, the data are the black lines. These are examples of populations of neurons in different, both of the monkeys for the cylinder task here. But if you look at what the correlations between predicted and actual firing rates are, the models do quite well. So, okay, so we are able to use a simple linear time invariant model to reconstruct the activity. So what? Well, let's go back to our question. Cortical drivers. Here's what you cannot extract from raw data. So if you build a model, you can actually compute what's called the solution, x of t. What does x of t look like? If you just solve that difference equation that I showed, this is what you would get with a zero initial condition. It would just look like a convolution of a to the power t with bu of t. You're convolving these signals. So basically what that means, at any given time, the firing profile, the firing rates of every cluster obey this equation, okay? And that equation is actually powers a to the power t, powers of a. So if you're thinking of a being like this connectivity, be careful, because the firing rate, that connectivity changes with time, with powers of a squared, a cubed, over time steps. So what I want to call your attention to here is in our model, X1 of T are all the M1 population cluster, firing activity of M1, and the PM populations are in X2. And if you just look at the equation, I can decompose this solution as follows. So for M1, which is X1 of T, I can decompose it as this Z1 plus Z2. What is Z1 plus Z2? It's the contribution of M1 populations affecting itself. So how do my populations within region affect my activity? And Z2 is how much does PM drive M1 activity? And when I add the two contributions, that gives me the firing rate. Okay, but these drivers, Z1 and Z2, I can only get from the model, powers of A and B. Similarly, I can break down and look for who's driving PM. There's going to be a contribution from motor cortex, those populations, driving PM as well as PM driving itself. So let's look at some results here. Okay, when I think about that, the first thing I want to do, let's take this is a mallet task for subject 2, monkey 2. All I'm drawing here is the firing rates. So this is what you've seen before. PM is, yep.
We have about 1
minute.
or 2 minutes.
Okay,
yeah.
second to last slide. Okay, the second to last slide. So in this case, this is just firing activity, what you've seen before in the movie, some activation in PM and M1. But here are the drivers. So this is who's driving motor cortex. The black says PM drives motor. Wow, this looks like conventional thinking. PM is driving the activity in M1 more than M1 is driving itself, okay? And then when I look at who's driving premotor, actually premotor drives itself. So this is actually conventional thinking. But if you look at the sphere task, and I'll probably end with this, what we saw in our sphere task is the opposite. That actually, if you look at who's driving PM, this right picture here, M1's driving PM. The pink curve is above the green curve. So there's an actual task here during sphere where M1 is driving PM, okay? Not conventional. And what I'm going to do, due to time constraint, I'm going to show you the last result. If you think of those as drivers, those signals, who's driving M1, who's driving PM, and look at cortical drivers as a function of kinematics, you really extract that relationship, which means the more different the behaviors are across the monkeys, the more different the drivers are, okay? And so I'll end there. I'll let you read the conclusions if you like. Thank you so much for listening. Thanks, Ava.
Thank you so much.
Thank you. Should
you ask the question?
So I don't. Okay. One question. Is it possible to fit fluctuations about the mean deterministic behavior by adding a noise
so
term
implicitly,
into the
although it's not there, there is a noise term in the model. So what we end up doing when we do the estimation of A and B, we are implicitly adding white Gaussian noise to the model, which allows for the variations. And that's how we actually estimate A and B, assuming additive white Gaussian noise, independent of the initial neural state. Is that the question?
I think so. Yeah. Excellent.
Excellent.
Thank you so much. That was a great talk.
Thank you.
Thank you, Evie.
So I would like to say thank you to all of our speakers. Thank you for sharing your work and your diverse perspectives. And we would like to thank the organizers, especially Leslie Weeks, who put so much work and help into making this event happen. And the organizers of the conference, Surgeon and Anne-Marie. And we'd also like to thank the rest of the Diversity, Equity, and Inclusion team, which would be Eugenia Chiapta, Maria Geffen, Weiji Ma, and Eric Shea Brown. Thank you all so much for coming. Enjoy the rest of the conference.
Thanks so much. Bye-bye.